In this lesson, we begin to look at cell transport and specifically passive transport. In this lesson, we begin to look at cell transport, and specifically, we're going to look at passive transport. So our learning objective is that by the end of today's lesson, as you've seen already, you're going to be able to explain how passive transport works. And in this lesson, what we're going to talk about is the movement of materials in and out of cells, as well as moving materials in between cells, from one cell to another, for instance. And there really are two ways that this happens. It either happens with energy or without energy. So, out of the two terms, active and passive, which one do you think requires energy? Well, if you're thinking the active transport term sounds like the one that would utilize energy, you would be correct. And we're going to get to that in another lesson. Today, we're going to focus on passive transport. So imagine that you and 20 of your classmates are in a room together at school, and I, your teacher, grab a can of air freshener, and up by my desk, up at the front, I start spraying the air freshener in the air. Now, imagine me asking you to raise your hand once you can smell the spray. Well, at the beginning, the students at the front of the class are going to smell the spray. And as time goes on, students more towards the middle will begin to raise their hand, and then the students towards the back will begin to raise their hand. So what do you think is happening to the spray? Well, the answer to that is that the particles of spray are slowly moving out into the room. So how did the particles start out? Well, they started out all bunched together, and then they started to spread out. And this tendency that things have to move from being all bunched up to being very spread out is called diffusion. You see that word right there on our slide. And this is when the same particles of things are all bunched together and they spread out. So when the same particles of things are all bunched together, we call that a high concentration. And when there are only a few of those things in the same area, we call that a low concentration. Can you think of another example of something that's in a high concentration or a low concentration? Well, maybe you thought of the analogy I used before, where we're all sitting in a room together. If it's a small room and there's a lot of us, that would be a high concentration of us, right? But if there are only a few of us, that would be a low concentration. Let me ask you this. Back to the air freshener analogy, do you think that the air freshener needed any energy in order to undergo diffusion? Well, if you're thinking no, you're thinking correct, because diffusion does not actually require any energy in order for the particles to move, because diffusion is what we call a passive movement. It does not require energy. So it's diffusion that is the driving force behind the movement of many substances across the cell membrane. In fact, some materials like oxygen diffuse across the cell membrane without using any energy at all, but other materials would actually require energy in order to pass through. Now, on screen here, we do have some images here, right in this area here. And we have a cell membrane here. That's this track-looking portion of the image. And we've got some molecules on either side of the membrane. So underneath the membrane, we see very few molecules. So that would be considered a low concentration of those molecules inside of the cell membrane. However, on the outside of the membrane, up here, we see a high concentration of whatever those molecules happen to be. And so let's go ahead and label our image. So we've got, as I said, the cell membrane. We've got the area inside of the cell where we have a low concentration, and we have the outside of the cell as well. Now, those little spheres, the little blue spheres that are found in low concentration inside the cell and high concentration outside of the cell, are what we call solutes. The reason that we call them solutes is because they're dissolved within the solution. So they are called a solutes. That's an important word for you to know. Solutes, okay? So what do you notice about the solutes inside and outside of the cell? Well, I kind of already said it. There are actually more solutes outside of the cell than there are inside the cell. Okay, we have a higher concentration of solutes outside of the cell than we have inside of the cell. So what we would find happening is that we get the solutes moving across the cell membrane. Now, what do you think those black arrows that you see, what do they mean? Take a minute to think about these arrows. One points in, one points out, another one points in, another one points out. And they must indicate something, or they wouldn't be there. So what do you think it is that they indicate? Well, what we find is that in diffusion, we find that where there's a higher concentration of solutes in solution on either side of a semi-permeable membrane, which, by the way, our cell membrane is semi-permeable, which you should already know. But what we find is that when solutes are in a higher concentration on one side of the membrane than on the other side, those solutes will begin to move across the membrane to the area of lower concentration. This is called a concentration gradient when it moves from higher to lower. Now, the concentration could be higher inside of the cell, and therefore the concentration gradient would be from inside the cell to outside the cell. But in this particular illustration, we see that the concentration is greater outside of the cell, so the concentration gradient goes from outside the cell to inside the cell. Now, eventually, there's going to be more solutes inside of the cell than outside of the cell if they only go one direction. But we have arrows here that indicate that there is some movement of solutes the other direction against the concentration gradient. Now, basically what happens is solutions and little molecules, these solutes, they want to be in what we call equilibrium. They want to be at the same amount both outside and inside of the cell on either side of the membrane. So because there's no energy required, those solutes are pretty free to just kind of do whatever they want. And so they do kind of flow along the concentration gradient until they're pretty even on both sides. But then we have movement of solutes kind of coming in and some going out, and it just kind of fluctuates. So it's never exactly the same all the time, but it generally stays pretty well even once that concentration gradient has equalized. Okay. So once we get to equilibrium, again, we just have some kind of movement of some solutes moving in and some moving out. Now, what you might also notice is that in this second image, the one in the middle here, you've got actually three arrows pointing inwards and only one pointing out, indicating that there is more movement of solutes across the membrane to the inside of the cell than there are solutes moving out of the cell. And that's because the concentration is higher on the outside. So the concentration gradient would indicate that there's going to be more movement of solutes across the membrane towards the inside of the cell. Now, in this image, you see that there are two arrows going each direction, one pointing inwards, one pointing outwards, the third one pointing in, and the fourth one pointing out. This indicates that we're pretty much at equilibrium, and the solutes are just kind of moving back and forth a little bit as they will. Okay, so it doesn't have to be exactly perfect, equal amounts on each side, but it's going to be very, very close. And that's passive transport. So that's passive transport when it comes to diffusion. Now, there is a type of passive transport that we call facilitated diffusion. So in some cases, when diffusion starts to happen, the particles involved need some assistance moving across the membrane. And they usually need this help because the physical characteristics of the particle or the solute or the physical characteristics of the membrane are somehow preventing that free flow of solutes across the membrane. So facilitated diffusion is the process where molecules that can't directly diffuse across the membrane pass through special protein channels. Basically, you could look at them as tunnels. And although facilitated diffusion uses these special channels, it's still considered a form of diffusion. That's why it's called facilitated diffusion. It needs a little help, but it still does not require the use of any of the cell's energy. So the way that facilitated fusion occurs is there is a protein embedded in the cell membrane called an aquaporin. You see that highlighted here in our image. And in fact, water, which is made up of two hydrogen and an oxygen atom, use aquaporins in order to move across the membrane. Water can't get through the membrane on its own. This is partially because of the makeup of the cell membrane, which has the hydrophobic tails on the inside. So because of these hydrophobic areas, water has a difficult time getting through the cell membrane. So water can easily pass through an aquaporin, a protein embedded in the cell membrane. And this actually, when water passes through these aquaporins, this is what we call osmosis. So osmosis, by definition, is the diffusion of water through a selectively permeable membrane. These aquaporins make the cell membrane semi-permeable. So the trick in understanding osmosis is that osmosis is about the movement of water molecules, not other larger particles. And you can see some of those larger particles in this illustration. But these smaller molecules, the red and blue ones, those are water molecules. The green ones are some other particle of something. It doesn't really matter what it is. What's important is that it's larger. And you could pretty much tell just by looking that these larger molecules probably aren't going to fit through those aquaporins. And that indeed is the case. So if we were to label some of the things we see in our illustration here, we would see that we have an aquaporin. We also have water molecules, which I've already pointed out. And you've also got the cell membrane where the aquaporins are embedded. And what this other green particle is actually in this particular case is sugar. And that's important because we're going to use sugar in our next illustration. So we're going to talk about osmosis. And one of the things about sugar is that it is too large, as we already talked about, to actually move across the membrane. So by looking at this particular illustration, which side looks like it has a higher concentration of solute? And again, solute would be the non-water particles. So don't count the water, count the sugars, right? What we see is we have one sugar on this side and we have three sugars on this side. So that means the higher concentration of sugar, or solutes in this case, is over here on the right. All right, we also see some white arrows in here. And what do you think those white arrows in this figure represent? Well, if you're thinking that they represent the movement of the water molecules through the aquaporins, you would be absolutely correct. But why do you think it is that there are more arrows pointing to the right than there are pointing to the left? In other words, there are two arrows pointing towards the area where the sugar is in higher concentration, and only one arrow pointing to the area where there is only one molecule of sugar, which is the low concentration. Why do you think that is? The sugar molecules can't cross the membrane because the proper types of proteins are not present. Now, sugar can actually cross the cell membrane, but we're going to learn about how it's able to do so when we start talking about active transport. But when it comes to passive transport, there's not a way for sugar molecules, which are quite large, to passively cross over the membrane. All right, so here we have a test tube, and we've got sugar water in the test tube, and we've got a barrier, which you can see here. That's basically a semi-permeable membrane. So water is able to osmote across that semi-permeable membrane, that barrier, but the sugar cannot. And so we also notice here that, like in the previous image, we have a higher concentration of sugar on the right side than we have on the left side. We will also notice, you may also notice, that there is the same amount of water, or solution really, on both sides of this U-tube. Okay, U-tube. This is not a YouTube channel. But this is a U-tube, and it is a type of tube that we use for utilizing osmosis. And let's go ahead and label these real quick. So we've got lower concentration of sugar molecules on the left, higher sugar concentration on the right. And what we find happening is that in order to become equal, we have to get some water to move to the right side in order to dilute the sugar, so that by volume, by the amount of water and solution on each side, the concentration of solute becomes very, very similar based on volume. So we're really talking about percentages. So while there may be, let's say, 70% of the sugar is on this side, or the concentration is 70%. We'll say it's 70% sugar, or 30% sugar to 70% water. Whereas on this side, it might be only 15% sugar and 85% water. What's going to happen is the water's going to osmote or diffuse across that semi-permeable membrane until the concentration, or the percentage of solute to water, is about the same on both sides. So let's just say now it's 50-50 water to sugar and 50-50 water to sugar on this side. So basically, the net movement of water molecules is to the right side in this illustration, because initially the concentration of water is lower there compared to the sugar. And the movement of water molecules across the barrier actually changes the water level on both sides of the tube. So the water will tend to actually move across the membrane until equilibrium is reached, and at that point the concentrations of water and sugar will be the same on both sides of the membrane. Once this happens, the two solutions will be considered isotonic. That's a very important word. And when the experiment began, the more concentrated sugar solution was hypertonic, whereas the diluted sugar solution was hypotonic. So those three words, isotonic, hypertonic, and hypotonic, are very important words for vocabulary terms. So make sure you know the difference between the three. All right, so driven by differences in solute concentration, the net movement of water out of or into a cell produces a force known as osmotic pressure. So we're talking about osmosis, right? So the pressure that's created by this movement is called osmotic pressure, pressure created by osmosis. So if we have an isotonic solution, well, let's first talk about what that means. Well, isotonic means that the concentration of solutes is the same inside and outside of the cell. So which way is the water going to flow? Well, if you're thinking, if it's isotonic, if the concentrations are the same on both sides of the membrane, then the water molecules should move equally in both directions, and that would be correct. Okay, so we've got water in and water out. Now, this is the case in both animal and plant cells. If we had a hypertonic solution, what would that mean? Well, that means that the solution has a higher solute concentration outside of the cell than inside the cell. So in this case, which way would the water flow? Well, there would be a net movement of water molecules out of the cell. And so if that were the case, what would happen to the cell? Well, if you're thinking that cell is probably going to shrink or shrivel up, you would be correct. In fact, that's what we see happening here and here. And in fact, that's what happens when plants get all wilty looking. It's because they don't have enough water inside of their cell. All right. Now, the third possibility here is hypotonic. And this, of course, is where we have a lower solute concentration on the inside of the cell. Nope, on the outside of the cell. Okay, so when we have a hypotonic solution, we get a net movement of water into the cell, and this causes the cell to expand. It's all swollen up. So the differences between plant and animal cells is that animal cells could actually burst when they're in a hypotonic situation. And while cells with a cell wall might be able to maintain their shape because of the presence of the cell wall, the higher osmotic pressure does weaken those plant cells, even despite the fact that there's a cell wall. So think about when a lawn care company sprays a fertilizer and water mixture onto grass. If they use too much fertilizer and too little water when they spray the grass might actually die and the lawn will turn brown. But would the grass cells with a high concentration of fertilizer have gained or lost water? Well, they would have lost water, and this is what would cause the cells to die. So which plant cell in this figure shows what would happen to grass at the cellular level? Well, if you said the hypertonic solution, you would be correct, because the fertilizer water mixture is actually hypertonic compared to the cytoplasm in the cell of the grass. And that, my friends, is passive transport. In the next lesson, we'll learn about active.